Okay, so hello everybody. I can see we've got something like 27 people on the call at the moment. And let's just wait a little bit and see if we get any more numbers. It looks like you've all managed to find us okay, 29 now. Up to 30. Okay, and I think maybe one of the first things that we can do, just whilst people are perhaps starting to join us, is I can make sure that I've got the other sort of main panelists who are going to be talking on this part of the workshop with me, where we're going to be looking in particular at ozone, pests and diseases, heat and drought, and evapotranspiration. So if I run through the people that I think should be here, do we have, let's see, Jonas Jägermeier? You out there somewhere?
Yeah, I'm here.
Hi, that's nice to see you. Hello. Very good.
Good to see you all.
What was that? Sorry. Okay, and then I think we've got a group of pest and disease people. Letitia, Simone. You want to just unmute and say hello, just so I can make sure you're there?
Yes, we are.
Hey.
And we've got a nice bit of feedback as well. I think that might have been Letitia.
Yeah.
Okay, right, good. We'll see how that works. And then finally, we've got evapotranspiration, who is Bruce.
Hello.
Hi, I can see you there, Bruce. Great. Okay, so I think that means that we have the main people who are going to be talking at some point. So I think one of the first things we need to do is decide who would like to rapporteur back on this session. I thought that would be a good thing to start off with first, so we don't forget. Any volunteers for reporting back to the main group? Everybody's gone very quiet. Simone, is that like you might be about to volunteer?
Only if I am forced to do that. No, yes, I can try. Yes.
Okay, that would be great.
Thank you for the opportunity.
Yeah, I think it would be useful if there's just somebody else, because I'm going to be struggling enough with the technology here. So thank you for offering that. Right, so I have just a few sort of introductory slides to try to give you a sense of what I think we should be talking about in this session, and then we'll go through each of the other different types of stress, if you like, and then by the end of those presentations, hopefully we'll have a good idea of what we all think we should be talking about in this session, and then we should have something like half an hour, I believe, to just discuss things so that Simone knows what he needs to be reporting back to the main group. So I'm going to try just get back to the right slide, and I'm going to try and share my screen. Hopefully you can all see a screen.
Yes, it is clear.
Good. I'll just try and with a PowerPoint presentation. Okay. So, right. So this session is looking at advances in environmental stresses and modeling of those stresses. And I've just identified, how many do we have there? 5 different points that I think, personally, we might want to think about, but there might be other things that other people who will talk will also raise as well. But for me, I think the aims of this session are so that we can get a better understanding of why we're interested in thinking about ozone and pest and disease, heat and drought and evapotranspiration. Why are they important? And how can AgMIP potentially help us to understand them better? How can we start to think about these stresses by modeling them globally? One of the things I'm quite interested in is thinking about some of the interlinkages between these stresses. I'm going to talk a little bit more about the ozone stress, but I know from my experience with ozone, that there are a lot of interlinkages with those other stresses. So whether that's something we can start to think about, that might be quite interesting. And then also I think how these stresses are likely to be affected by climate change. Obviously climate change is having quite a substantial impact and causing these stresses to be even more enhanced than they would be otherwise. But thinking about that would also be useful. And then coming back and thinking, what sort of things could AgMIP be doing that could support this kind of stress modeling research? Okay, so I just thought it might be interesting just to show this slide, which is a study which was published back in a couple of years ago by Mills et al. 2018. And it sort of quite nicely brings together a number of these different stresses that we are talking about. So just running through the slide here, we can see that we have ozone, pest and disease, nutrients, heat and aridity. And this study used something called a yield constraint score to try and make an assessment of the constraint to yield by each of these different stresses. And we have this yield constraint score, you can see down here, going all the way from 1, where there isn't really any constraint, up to 5, where you would have quite serious constraints to yield. So just as an example, that 5, where we were actually able to assess yield losses based on these stresses, then that category of 5 was categorizing yield losses above 40%, so quite severe impacts on yield. So we get a sense then of across the globe where these different stresses are having most impact, and we also get a sense of the potential magnitude of these stresses in comparison to each other. If we look at this top left-hand panel here, we can also get a sense, if we start to make some sort of overlay of these stresses, we can also try to identify some particular hotspot regions, which are going to be at some substantial damage from a combination of stresses. So I guess one question here to people is, can AgMIP enhance this type of modelling assessment? This was obviously in quite early days of really trying to understand these different stresses and place them in some sort of context in relation to each other. So can we do better in terms of modelling these assessments? Can we improve those global estimates of yield constraints and losses? And can we think, start to think a little bit more about some of the interactions between those stresses? And then one of the other things that we also started to look at in this paper was, okay, if we know that there's a stress such as ozone stress, we know that there are certain physiological traits that you could introduce into a plant. So here we have an idea type, which would be an ozone tolerant crop, for example. So we know that those traits would start to improve that crop from the point of view of ozone, but can we also think about whether there would be synergies and trade-offs for some of these other stresses that we're seeing? So I just want to run through just a couple of these. So you can see that we split this out into traits that you could bring into the panicles, into the leaves and into the roots. And if we see here that anything that's given a plus here would mean that you have an ozone tolerant trait that actually gives you a benefit for other stresses. So we can see here, for example, improvements in seed quality and days to maturity could help with most of those other stresses that we see, so drought, heat, nutrients, pests and disease. If we're thinking about leaves and we're thinking about traits that might reduce the amount of ozone being taken up by the plant or the effect of that ozone once it's inside the plant, then we can also see synergies there for drought tolerance as well. And then for roots, traits that might enhance root allocation and root dynamics would also potentially have benefits for these other stresses. And just to flag up this one, pests and diseases, the right-hand column there, quite uncertain in terms of what the influence of some of these traits would be on pests and diseases. So that's something else I think that's quite interesting. So I think one of the sort of benefits of doing this sort of work, again, might mean, you know, can we within AgMIP, with all the knowledge that we have, enhance this type of modelling assessment? And then could this type of assessment actually be used to inform crop management, to make crop management better, and also crop breeding practices as well? Okay, so that's really my introduction, aside to say that this is the agenda that I'm planning for this session. And I hope I've got my timings right. If there's somebody else out there, perhaps Cynthia can just make sure that these timings are going to work and we're going to get back into the main room on time. One of the things I just want to try to see if it might work. So you can see that we've got this discussion area down here. I'm just going to come out of this for a second and see if I can share with you. I'm going to try and put this in the chat. Oops. Here we go. Right, so there's going to be a link to a Google document coming to you in the chat. And I'm just going to move that out of the way, move this up. Hopefully you can now see a Google document. Scream
Yes.
if you can't. Good. I'll scream if you can, even better. Now, I don't know if this is going to work, but this is your opportunity. If you can get in here, and I probably need to make sure, yeah, anybody with the link can access it, so that should be okay. Then you'll see some of these questions that are written down in here. So, for example, why is AgMIP well positioned to explore ozone? And these little red things represent post-it notes that you can grab and start to write stuff on as you're listening to things. So I think if people can go in here and feel completely free about adding bits and pieces of information, that would be absolutely fantastic. Then we've all got a resource that we can use and share.
Lisa, at least I don't see the link yet. Did you paste it in the chat?
Let's have a look. Did I send? Because I might have just sent it to one person. Let's try that again. Okay.
Now we see it. Thank you.
Brilliant. Okay, so the next thing is going to be whether you can get in. Hopefully you'll be able to. Yes, I can see people coming in. Excellent. Okay, so we've got these different questions. Why is AgMIP well positioned to explore these different stresses? What are the key interlinkages between stresses? So here you've got different matrices with a stress down the side and then your other stresses along the top. So we've got a couple of those, looking about those interlinkages. And then how can AgMIP provide support to advanced modelling for these stresses? So there's a couple of post-it notes available down there. So it would just be interesting if people can start to fill that out, and that might make mine and Simon's job a little bit easier when we report back. Okay, so that's that. I shall pop that over there. And then I think, unless are there any other questions before I launch into
Lisa,
yes?
sorry, just technical.
That's
at least
fine.
when I got there, I had view only permissions. So I don't know if you can make it like anybody with the link can edit, not just view.
Yes, I think I should be able to change that. Thank you for, I don't, it always does that. Okay, Alex, do you want to try writing something now and see if that works?
All right, let me, I think I have to reload it. Yep. All right, I just put something on slide 2, so it seems to be working. Thank you. If people haven't done that, you might have to close the tab and open it again.
Or just refresh. That does it to me.
Okay, great. So it'll be interesting to see what we get coming out in there. Okay, that's brilliant. Thank you. Any other questions about the introduction? Anything that I've missed that we should have mentioned in terms of what we're trying to do here? Alex again and Cynthia, I don't know whether there's anything that you wanted to add from an AgMIP perspective.
Nope, full speed ahead.
Great. Okay. Right. So I'm going to give just a little bit more information about ozone. And I'm going to try and do that in probably slightly less than 15 minutes. So hopefully you can see these slides again, and we'll go straight into ozone. So first off, I just wanted to provide a bit more information about what the effects of ozone are on agricultural crops. So we have visible injury. You can see a visible injury image there. Hopefully you can see my cursor moving around. We also have biomass losses above and below ground. Down here you can see different sizes of wheat plant that have been grown just outside Lahore in Pakistan. This one has been grown in ambient air, and it's thought that it's the ozone that is causing that reduction in the growth of the plant, compared to this larger plant, which has been grown in a filtered chamber where all of the pollution has been taken out of the air. And then those kind of growth losses translate into yield losses. So those are some of the key impacts that we can see. And then just to mention those open top chambers at the top, the sort of experimental apparatus that we use to test and find out what these sorts of impacts are. But then we also have other physiological impacts caused by ozone, so changes in photosynthesis, monoconductance, highlighted transpiration, because that's one of the other stresses we'll talk about here, changes in leaf area index, changes in leaf quality, so nitrogen or protein content of the leaves, changes in carbon allocation, so you have a tendency to find less resources put to the roots than to the shoots, and then also changes in reproductive ability. And then I think the other thing to bear in mind about ozone is that it's also a greenhouse gas itself. So when you have high concentrations in the atmosphere, then it's likely to be affecting near-term climate change. Okay, just to give an indication, so you've probably got a sense of this from the previous slides looking at those yield constraint scores, but this gives an indication of the size and the location of those yield losses that we might see globally. So this is for wheat, so these are production losses due to wheat, for wheat due to ozone. And if you take averages here, then we find that you get a yield loss of around about 10% in the northern hemisphere and around about 6% in the southern hemisphere. And what we find is that the humid and irrigated locations are actually most at risk, because those conditions will tend to increase the stomatic and the uptake of ozone, and therefore the damaging effects of ozone on the plant. And we can make economic loss estimates of those production losses, and they're quite substantial, about $24 billion each year. Okay, so that's yield losses, but there are other effects that we see as well. So we have reduced fertilizer efficiency. So this is because the fraction of the nitrogen that ends up in the grain is negatively affected by ozone. So that will reduce your fertilizer efficiency. It can also cause leaching of any surplus nitrogen that you might be putting on to your crop, and then that can also cause eutrophication. We also have drought tolerance being reduced. So here we can see what would normally happen. You have a drying soil. The roots from that soil would produce the hormone abscisic acid, which would then lead to the stomates closing and conserving water. Under the effect of ozone, stress ethylene is produced, and that actually inhibits this ABA hormone, which means that the stomates can't sense the need to shut, and therefore we start seeing an increase in water loss. We can also see effects on forage quality. So here, as we've got an increasing ozone concentration, or flux in this case, you actually reduce the clover fraction of grasslands, so you make them less productive in terms of their forage quality. And then we also find that ozone within ambient ranges reduces nitrogen fixation in legume species. So that adds a double whammy to that impact. And then finally, pests and diseases. So here we can see again this increasing in ozone concentration, and where we have ozone sensitive plants denoted by these closed circles, we see an increased rate in spider mite reproduction, which is a pest. So we can see interactions here as well. Okay, and I showed this previously. So this is, here we have our ozone impact assessment for wheat again, and you can see how the sort of geographical spread of that and the magnitude of that sort of within this 1 to 2 category. And we can compare that maybe with something like heat, which has a sort of similar stress, maybe slightly higher spatial extent of the stress, but then you can see heat is really getting up to these very high yield impacts. And so this kind of modeling that we did before is really just looking at those stresses individually. So it would be interesting, based on that previous slide where I showed how ozone, just for example, is causing interactions between these other stresses, to think about these interactions a little bit more. And obviously there's going to be interactions between heat and aridity, heat and pests and diseases and so on. So can AgMIP help us to understand that? Okay, and then just to think a little bit about future projections of air pollution. And again, this is ozone. So here in this top graph, we've got surface ozone changes. So from 0 indicated here, and then we've got this for 4 different regions across the globe: North America, Europe, South Asia, and East Asia. And then we have these different RCP scenarios. So if we look in particular at South Asia and East Asia, in South Asia, pretty much all of those scenarios are going to see an increase in ozone concentration from 2000 up to sort of 2030s and 2040s. In East Asia, you're seeing 2 of those scenarios at least, suggesting that you're going to have an increase in ozone concentrations. So what our ozone concentrations are going to look like in the future are really going to depend on the emission controls of those ozone precursors. And then just one other thing to point out. So some of the work that we've done has also been looking at the date and the release of different cultivars. This gives an indication of soybean cultivars. So this provides the slope of a dose response function for soybean. And that's the dose of ozone versus the response, some sort of yield. As you have a more negative number, then you're seeing a greater yield loss. And so what this is showing you is that as you have the more recently released cultivars of soybean, you're starting to see that they're actually more sensitive to ozone pollution. And we believe that's because those cultivars have been bred for increased gas exchange, which is great for photosynthesis, but then it also allows an increased uptake of ozone, which then causes damage. Okay, so what are we trying to do within AgMIP and our ozone initiative in particular? So we've been collating and standardizing ozone pollution fumigation data sets, and we've now set up an AgMIP ozone data hub where different crop modelers will be able to access these data to test and evaluate their crop models. We've established a group of ozone crop modelers. So some of these are people that have been just dealing with ozone and are branching into crop modeling. Most of them are crop modelers who are starting to bring some sort of ozone impact assessment into their crop model. Then we want to develop means to evaluate those ozone crop models using those empirical data sets. We'll be developing crop modeling protocols for global application, and we will then hope to conduct a crop model ensemble exercise so that we can understand the impacts of ozone both under current and future climates. And once we've done that, we should be able to assess the benefits of different emission reduction pathways for ozone. And that should help us advise policymakers in relation to food supply. Okay, and this just gives an overview of those data sets. So you can see the kind of spread that we have of those empirical data sets, a lot from Europe, some from South and East Asia. And then you also get a sense of the data sets and where we are in terms of checking and calibrating and which of those data sets are useful. Okay, and then I just wanted to spend a few minutes thinking about also what we can do within AgMIP Ozone to identify different research opportunities. So this, referring to this as our COVID-19 paper, but we sort of realized, as did many other people in the air pollution community, that obviously during lockdown we've had quite significant decreases in the emissions of certain air pollutants. And we believe that this could lead to a decline in seasonal ozone concentrations, which could then have some sort of positive impact on crop yield, at least during this 2020 growing season. So this offers the opportunity to test and improve crop models and for us to think about the experimental and exposure response relationships that we might be using, and to be able to do this under real world conditions. So just to run through what we can try to do here, we've done some preliminary work here. So this just shows you the difference in NO2 columns between March and April of this year compared to the same time last year. Those blue areas give you an indication that the NO2 column concentrations have decreased quite substantially. So that's in Europe, Asia, and the US. And then what we can also do is think about, well, how would those emission reductions or emission reductions similar to the size that we've seen over 2020 actually lead to changes in ozone concentration? So I just show this here. So these are our NOx, and then we've also got non-methane volatile organic compound emissions as well. And we can see that if we have these different scenarios here, which we've run through a model which gives us an idea of what the ozone concentration would be, we've used different emission reductions across different areas. So we've got 30% emission reductions in Europe and then globally. We've also got scenarios of 50% in Europe and globally, and then we've also looked at reductions in emissions from shipping and aviation. So we can run those emission scenarios through a simple model, and that gives us an idea of how this is our May, June and July ozone concentration would have changed in the year 2020. If we have this data, then we can bring that together to form a 3-month AOT40 metric. So this is a metric which allows us to get an idea of the impact on crop yield. And just to show that here, so this is a dose-response relationship. We've got our AOT40 dose of ozone here, and we've got our relative yield up the side. So we can look at the difference in the AOT40 or the dose, and then we can read off a difference in our relative yield. And just some very preliminary results using this fast tool that we had. We can see here that we've got different countries, and we've got the yield increases that we would expect to see, because now we've got a reduction in the ozone, so we should have more crop production going on. And I've just picked out Spain here, so these different bars give you an idea of the emission reductions that we have if we just focus on emission reductions in Europe versus the world, versus those coming from shipping and aircraft, and then versus that if we combine all of those different emission reductions together. And so in theory, we might be expecting to see, just for that country in Spain, that you might see yield increases of somewhere in the region of about 6%. Okay, so that's just some work that we are thinking we might be able to do, and it might be great to be able to use AgMIP to allow us to do that work. These are the things we'd need to be able to do it properly in a more robust way. So we'd need to be certain about those emission changes in 2020, then how those emission changes convert into ozone concentrations. We'd also ideally get some on the ground stats of agricultural yields and how they might be different in 2020 with previous years. And then we'd have information that would allow us to really assess these different methods, including crop modeling approaches, to understand the impact of ozone. Okay, and it would be great if AgMIP could help support this activity. So that, I think, just gives quite a nice example of the sorts of things that we might be able to do with support from AgMIP. And with that, I can stop and just thank all the people that have helped with this, some of whom I think might be on this, in this workshop. And then I think I can hand over to Letitia and Simone, if you are out there.
Lisa, shall we hold our questions for the end?
Oh yeah, well actually, if there are questions, maybe we could take a few questions if there are some out there. That would probably be good.
Fast ones if there are.
Yeah, thanks, Alex.
I had one, since I did interrupt you here. I don't know if there are others. But could you just go up one slide to your, or I guess slide 18. Yeah. Could you just help us, point of clarification, you have two y-axes. Could you just describe what those refer to?
Okay, so this is just giving you an indication of the size of the emission reduction. So for example, here, this kind of orangey area is just a 30% emission reduction, and the redder area goes all the way up to a 50% emission reduction. That would be globally for NOx and VOCs.
All right, and then can you see the yield increases? That's that side.
That's that side, that's the yield increases. So we're seeing sort of essentially yield increases of around about 6 to 7 at the highest end.
Thank you.
Okay, great. Oh,
This is Lily. I don't know, should I hold on after all the presentation or just ask you right now?
no, just ask for now.
Okay, I'm
not feeling
curious.
too bad with time.
Okay, I'm curious. For this slide, this is your prediction for how this ozone reduction will increase wheat yield. But how are you going to compare it with the reality? I mean, this year there are a lot of countries reporting yield decrease because of lack of labor and also the extreme events like in China, the flooding and drought there. So how are you going to really compare those estimation with the real data?
Yeah, no, it's a really good question. And it sort of comes back to what we've got in this slide here. One of the key things that we need is to start to collect the actual on the ground data of agricultural yield. So we're working with JRC, which is one of the institutes that actually monitors wheat yield production across Europe. And I think in the first instance, we'd have to have a look and see, well, what has actually happened in terms of those wheat yields and try to get an understanding of why. When we first wrote this paper, at least in Europe, it was felt that COVID-19 wasn't having other impacts on wheat production in particular. So at that time, we thought that it might be possible to see this ozone signal. But you're right, we need to really understand what other impacts might have been felt on things like wheat farming over that time period. The other thing maybe also just to mention is because ozone is a secondary pollutant, it's not always so straightforward to know whether it's going to go up or down as the emissions change. So that's something else that we need to look at. But we kind of saw that the opportunity is potentially out there to perhaps do something interesting, maybe focusing on particular locations across these different regions where we don't have some of these other confounding factors. Any other quick questions? If not, I think it might be time. And remember to keep filling stuff out in the Google sheet if there is anything that you want to raise or mention about this. I shall stop sharing my screen and hand over to Simone and Letitia, I believe.
Yeah, I will try to share my screen. Okay. Can you hear me?
Yep, we can hear you very well.
Okay, great. And can you see the screen?
Yes.
Okay.
Yeah, great.
Great, thanks. So this presentation was prepared together with Simone Bregaglio from CREA in Italy, myself and Serge Savary, and both of us are working at INRAE Toulouse in France. And so I will start to provide some general background information, and then Simone will in more detail present to you the latest progress we made in this group. So this is the first background slide about pests and diseases, and it's an illustration with the work which was done recently on wheat, rice, maize, potato, and soybean, which enabled from a survey made with experts globally to provide estimates of yield losses, both from individual and combined diseases and pests. So you see on the screen on the first block is about wheat, then rice, maize, potato, and soybean. And what we can see, what we can see also within each block are different what we call food security hotspots. So the first column is about global yield losses, and then you've got US and Canada, South Brazil and Argentina, Northern Europe, China, the Indo-Gangetic Plains, Western Africa, and Northern Asia. Western Asia, Northern Africa, and Sub-Saharan Africa. What I will not go in details, but what you can see is that when we talk about pests and diseases for a given crop, the main diseases and pests would be anything between 10 and 20. And these are the names on the columns. What we can see also is that the patterns, if you like, or the profiles can depend a lot depending on the region we are talking about. So in the total, 137 pests and pathogens were reported in this large study. And if you want to look at it in more details, it has been published last year in Nature, Ecology and Evolution. What we could do also is look at the combined losses from pests and diseases for each crop, and then by food security hotspot and globally. Okay, the message from this slide, and it's one of the key messages we'd like to also give to the other groups of epidemics, that, well, we can see that the crop loss ranges across food security hotspots it can be very large. It's from 8 to 41%, so that can be very large variation. And importantly, these ranges of yield losses imply that simulated yields, which do not account for diseases or pests, would represent overestimates. We feel it's an important message to send because usually it seems that it is really overlooked. In a few articles, people would really speak about attainable yield or potential yield, but very often the articles would report simply yield, and it's very important to have a clear idea of what kind of yield level we are speaking about. Now I'll come back to the background on AgMIP. So its mission is to improve agricultural models for pests and diseases and enhance the scientific and technologies capabilities to assess impacts of climate variability and climate change and other driving forces of agriculture. Labor was indicated earlier. This is one of the important also driving forces in agriculture, and there are others. AgMIP is an open initiative which is gathering scientists with a specific interest in the development and the application of simulation models of agricultural pests and diseases, and also their coupling with crop models. And I'll come back to that in more details. So when it's about modeling plant diseases, we can deal with many different approaches, different objectives. But we can identify really two main groups of objectives. The first one is about modeling in order to analyze and understand epidemics. So this would be more about ecological, an ecological field, if you like, and is dealing with really population dynamics. So this would be the dynamics of population of lesions in the field or globally, or population of insects. Then the second group of objective is about the analysis of crop losses. And of course, one or both of these objectives ultimately, practically, are aimed at improving disease management and reduce crop losses. So much progress has been made over the last years and even decades on the modeling of the effects of harmful organisms on crops through the concept of damage mechanisms, and Simone will come back on that. And as a result, it is now possible to model crop losses caused by one or multiple injuries in a generic manner. And this is despite the very limited availability of injury functions. And we'll also come back to this. That's to say, field data which are about the dynamics of epidemics are very, very limited, and also quite heterogeneous. So one thing we are doing both when dealing with modeling the dynamics of epidemics and also of crop losses is that we need to have a modeling framework for injury functions. So this graph on the left would represent if you like, the time course of an epidemic, and this can be used as a driver for crop agrophysiological models which are meant to simulate yield losses. Now, this disease progress curves will change in magnitude depending on the conditions, maybe weather conditions or other conditions. And then there will be several diseases which are affecting a given field. So we need to account not only one disease, but several diseases which can have very different shapes because their ecological features can be very different from one disease to another. And so one strategy which can be used is to represent what we call injury scenarios. That's to say, if you like, the time course of crop health, which would include the dynamics of several diseases at several levels. So this is one approach we want to implement when dealing with identifying injury drivers for crop loss models. So there are two main components, again, of plant disease modeling. One is about the epidemiological modeling, and the other is about the generic crop loss modeling structures. And today we will report only on crop loss modeling. So the general framework we are using for crop loss modeling starts with a very important concept, which is the concept of production situations. A given production situation is characterized by several factors. So biophysical factors, weather, radiation, temperature, rainfall, soil characteristics, and also cropping practices, which are actually the adaptation of farmers to the other environmental conditions. And they include crop establishment, nutrient, and water management. And this a given production situation will, if you like, be used as the main driver of a crop growth model. And so for this, we will calibrate or identify a set of vector of parameters, and mainly the RUE and also the relative rate of leaf senescence, in order to simulate the attainable yield for a given production situation. So if you like, a given production situation relates to proxies, which are input in the model as parameters, which then allow to simulate the attainable yield, that's to say the yield of a crop without any injuries from pests or diseases. On the other hand, production situation is a very important concept because it is strongly linked, and this has been shown in different systems, mainly rice in tropical Asia, but also wheat. So production situations are driving what we call injury profiles. So an injury profile, if you like, is a combination of the different pathogens, insect, pests, and also weeds, which are affecting a given crop. And so injury profiles and production situations can be used as drivers for a crop growth model into which we include damage mechanisms. A damage mechanism, if you like, is the insertion into the model of the physiological effects of injuries on the growth and resulting yield. And so this allows to simulate the actual yield. And then the difference between attainable yield and actual yield is yield loss. So this is a general strategy we are using. So I think this slide could, and Simone will use it later on, can be also a bridge between the pest and disease epidemic group and the groups which are dealing with other stresses, and also with other groups which are dealing with potential and attainable yield. We find very useful to use the concept of production situation, which is, if you like, embedding the different defining and limiting factors. And so it allows us to look at these factors not individually, but in a combined way, which we find very efficient. And now Simone will just explain to you what we have done recently in terms of crop losses in a work we did with several scientists on wheat.
Thank you very much, Laetitia. May I ask you to continue to share your screen, please, so we don't change? And
Sure, yeah, no problem. Go ahead.
Thank you very much. So the last part of this presentation deals with the work that we did with the group, which is part of AgMIP, but also of another project in a EU project, MAXUR, which is actually very linked to AgMIP in general. So here we realized the concepts that Laetitia just explained. So we started from a reference model, which is called the wheat pest, which was developed by Serge and Laetitia. And we, let's say, transfer the knowledge that was implemented in that model to other 4 wheat models. So we establish a protocol to, let's say, to equip the crop models for the simulation of pests and diseases. So the main objective of this work was to make this advancement, because nearly none of the crop models can do that, and also to assess the different sources of uncertainty in this kind of study. Next slide, please. Yeah. Okay, so this is just a brief overview of the simulation protocol that we have developed. This paper is under a major revision, and this is my task. I'm very slow on that. Sorry, Letizia, and also Roberto Ferrise. I take the opportunity also to present the other components of this group. So Roberto, then there is Christian Kersebaum from ZALF, who contributed with the Hermes model. Then also there is Willington Pavan and Sentol Dasseng, who join us with the DSAT and WIT model, and we are. And that's okay. And then that's me and Tommaso Stella, who joined with the WOFOST. So we have very different models, which simulates crop physiological processes in a very different manner. So we tested them as coupled with the different disease. We focused on wheat as a case study for wheat diseases, foliar wheat diseases, so powdery mildew, leaf rust, yellow rust, and Septoria triticum. We tested 2 different shapes of the disease. So before Letizia presented you which kind of shape of injury profile a disease can take. Here we tested just 2 and different level of maximum disease severity. All these factors were tested in a factorial combination in order to assess with an analysis of variance the magnitude of their effect on yield losses. Then we also tested different level of calibration, so low calibration and high calibration, in order to see if the models were able to reduce the uncertainty when more reference data were available for their calibration. So next slide is about the very short results. So we, with this ANOVA, we were able to assess the relative contribution of the different factors. And we saw that the disease level has got the main importance. So the level of disease severity, so the percentage of host tissue affected by the different diseases. Then the model, so the 4 models have had a very large effect. The main message here is that there is an overall agreement between models, but the choice of the models can lead to a very large variability. So the next slide is just the conclusion of this work in which we realized for the first time this coupling, and we extended to other crop models. In the paper, which will be hopefully accepted, we will also release a protocol for other crop modelers to perform a similar work. We are open as AgMIP initiative to address, let's say, the questions from other groups if they want to perform a similar work than ours. And then the other slide is just thank you, Letizia, the framework of activity. So our aim, and also Serge is here, and is to develop this crop health group in which many models can join and share these efforts, because we believe that if this coupling between disease and crop model will be realized, we will all benefit from different perspectives, as also Lisa mentioned in the introduction. So I think that in the very last slide, and I'm concluding, there is just a hint on how AgMIP can and AgMIP could foster this collaboration, because it's also true to say that in the last years, this has not been really realized operatively. So it was like a spot initiative, what we did in the work that I just presented. So here you see the same workflow that yes, overview that Laetitia gave before. And with these yellow arrows, you can see where AgMIP can really join this PDMIP initiative. If we are able to collect different modelers, different crop models, and they want to join this effort, we can provide the rationale to include the damage mechanisms into their model and to perform a parallel simulation of attainable yield and actual yield in order to have better realistic simulation of yield losses globally. And that's it. Thank you very much.
Okay, thank you very much indeed, Simone and Laetitia. That was a really nice, clear presentation, and it's really interesting to see where you think other crop modelers could work with you to try to get a better understanding of these pest and disease impacts. So I think we've got a little bit of time for just some initial questions direct to that. Is there anybody that would like to ask a question? I can see Jonas. Yes.
Oh, cool.
Please.
Yeah. Cool. Yeah. Thanks for that. That's really interesting. Great to see yet another MIP on that front. No, I think I have two questions actually. So one is, I think when it comes to the impact modeling, the mechanisms are one aspect, but I wonder how you model an outbreak. Like, when would a certain disease hit, or would you only look at average conditions over time? I don't know, that's just a general question. And then the second question is, if you see a way to incorporate your work, make it available for, say, our group, the process-based modeling groups, which, you know, you already incorporate some of the models, but do you see a way to produce a layer, kind of a product that we could use in post-processing for climate change projections, and say, over time, you provide us with a map of likely percentage impacts that we could include in our analyses and therefore harmonize our efforts? That would be a really valuable thing to use. Thank you.
Hi, this is Serge on the phone. I'll try to answer the both questions, if that's okay with Simone and Laetitia. With respect to the first point, what we do as plant disease epidemiologists is make categories between chronic, emerging, and acute epidemics. And those temporal patterns are very different. They are also spatially different. So we don't address them the same way. And in essence, this is captured by the injury profiles that Laetitia has been showing. And so in essence, we would address these different types of epidemics the same way, but of course, the dynamics over time would be different. And so we mechanistic simulation models at the field scale or at the plot scale are unable to address the spatial pattern of epidemics, which does exist and is very important. So this would be a long discussion to address, but yes, your point is very true. It's very important, and we do make a difference. For your second point, well, we have, over the years, we have generated a number of very simple mechanistic simulation models. The first of them was rice pest, and rice pest was successfully linked with global weather data from the NASA, and it was also linked with epidemiological models to simulate the impact of epidemics and the consequences of epidemics at a fairly large scale. It's been done in Indonesia, it's been done in China. Some of this has been published, and so we could share the references with you. The same thing has been done for wheat to some extent. We've been able to link a very simple epidemiological multi-disease epidemiological model for wheat diseases for Western Europe and link this to European climatic data. And that allows us to map disease risks across Europe. But this is only disease risks. It's not providing measurements of yield losses so far. We can do that. It's doable, and we are working on it. The big challenge I see is several things. One of them was mentioned by Simone and Laetitia. The first of them is that we are dealing with many different diseases, many different pathogens, viruses, bacteria, fungi, oomycetes, nematodes, and we deal with different pests. It can be aphids, insects, spiders, spider mites, and so on and so forth. In rice alone, we are dealing with at least 30 damaging pathogens and pests. And so it is very difficult to develop a single platform where one would be able to simulate the dynamics of this entire zoo, if you like, and then link this to a crop loss model and so on and so forth. And this is why the point made by Laetitia regarding production situations first, injury profiles associated with those production situations next, and then dealing with scenarios, this now becomes manageable and that's feasible. These are the challenges we face. And it will be the same for, we've worked on coffee, we've worked on groundnut in West Africa. There is work on different crops. There is a number of examples to show that this is a reasonable way to approach the problem.
Okay, that's great. Thanks ever so much, Sergey, for giving such a comprehensive answer to that. That was really, really useful. I think probably in the interest of time, we should probably go on to the next short talk, which is actually Jonas, I do believe. I hope you're ready for this. And you're going to talk about heat and drought. So, excellent.
All right, thank you, Lisa. I believe you can see my screen?
Yeah, can see it.
Awesome. Okay, I'm Jonas Jägermeier. I'm with Columbia University and NASAGIS, closely working with the GGCMI team, the Global Gridded Crop Model Initiative within AgMIP, running process-based models in different contexts. And today I would like to take that opportunity and look at the responses of these models to future extreme events, and in particular droughts and heat waves. This is work in progress since we're aiming to use new AgMIP8 climate forcing, and the crop model simulations are just about to come in. So part of it is actual results. Others are proxy or proof of concept results. I will point that out, but that's why we have this flag preliminary here. But it's a good time to talk about this because today, October 13th, is indeed the International Day for Disaster Risk Reduction. I'll come back to that second, but just wanted to point that out. Now, we're interested in droughts and heat waves in particular because they can disrupt the food system. They can affect trade dynamics. They can affect price spikes or cause price spikes, and they're known as one of the more costly extreme events. Both droughts and heat waves are expected to become more frequent and more intense in the future. But we don't really know what the responses of global stable crop production to these events currently is and will be in the future. So there's a lot of question marks associated. Now, before we jump into that, I would like to throw in 2 slides on the most recent data we gained from AgMIP6 Insights, looking at long-term mean impacts, just to give some context and background. So on this slide, sorry, the animation doesn't work. On this slide, it's an average end of season global crop production impact simulated by an ensemble of crop models and climate models. Now, those are AgMIP6 bias adjusted and downscaled models, 5 of them selected, harmonized through the AgMIP framework. Some of you might know that. And we have so far 6 global crop models running these. I would like you to focus on the maize part first before we look at the others. I was meaning to mask them, this is not happening. So the colored bars are the mean impact for 2 RCPs. In green is the RCP 2.6, in yellow the RCP 8.5, and the bold black bar is the mean across all climate crop model combinations that are 30 in this case. Now we have a dashed and a solid whisker going across these, and that's the climate model range as a dashed line and the range across crop models. And that gives us somewhat of an idea of the uncertainty. Now we have 3 points I want to drive home from this slide, and then we move on. First is that maize is seeing larger and less and more certain impacts simulated, projected by the models compared to the other crops, followed by soybean with larger uncertainties. Wheat and rice are even less certain than that. All of these results are shown for the default CO2 case, and there are discussions related to that if the effects can be achieved at global scale. So that's another uncertainty not pointed out on this slide. But then the second point is the little gray stacks to the left of the colored ones is the previous simulation rounds. That's published in the Rosenzweig et al. 2014 paper using CMIP5 data. And we see that the overall ensemble is much more pessimistic now in CMIP6. And that has various reasons. First of all, the CMIP6 ensemble has a higher climate sensitivity on average than CMIP5, but we also have a different selection of crop models, with some of them are more pessimistic. But on top of that, the overall crop model ensemble is now more pessimistic than previously expected. The third message I want to have here is that the range across climate models increased now in the new simulation round, and the range across crop models significantly decreased. I just want to point that out. The one model, LPJML here, is the most optimistic model in our ensemble here, just because we come back to that model later, and I just want to point out that where it sits in context with the other models. Now, real quick, we can also look at time series and the concept of emergence. If we look at the historical range of data, this is the dashed horizontal line. We can point out a year when the mean, the smooth mean of projections would go beyond that point, or in this case, exceed the negative, the limit of the historical range, and call that emergence, and point out the year, which is 2036 in this case for maize at global level. We can look at that in different climate regions, major Krepp and Geiger climate zones with gains in the temperature limited regions, and major losses in temperate, subtropical, and tropical regions, with emergence sometime in the 30s in temperate regions, and as early as 2025 in subtropical and tropical regions, closely followed by the RCP 2.6 a few years later. We can also look at maps with major declines in global breadbasket regions, and the hatching indicates model uncertainty. So for maize, this is a pretty stark picture, large losses all over the place. Put this into a dashboard context slide, we see that wheat has much less of a signal with huge uncertainties associated. Soybean and rice have a somewhat similar picture with a peak and decline trajectory, first of a positive exceedance of the historical range, and then a negative exceedance in sometimes by the end of the century. Now, this is the context for looking at extreme events, because the average signal is one thing that we're interested in, but often it's the the yield variability, the year-to-year fluctuations, the impacts during extreme years that are especially important for food systems, for the farmers, for insurances, and so forth. Now, as I mentioned, today is the International Day for Disaster Risk Reduction, which is marked by a new report that was released yesterday by the United Nations Disaster Risk Reduction Framework, called The Human Cost of Disasters, an overview of the last 20 years. And they find a significant increase in overall disasters across the last 40 years. And it's especially droughts and heat waves, or they call it extreme temperature events, that have increased in number, droughts by about 30%, but extreme temperature events by more than 200% since the 80s, which is a dramatic number. Now we're trying to understand what are the impacts of these events on global agricultural productivity. We've tried to put these events into a model. In this case, it's LPJML indeed, and using this composite approach where we superimpose all of these events, we all put them together, normalize them, and have the average event signal in year 0 surrounded by the 6 neighboring years, and then average the signal to have this composite signal, and the little dashes along the y-axis are the standard deviations. Now, on average, heat waves in the historical period between 1960 and 2015, I guess, reduced global maize production by about 12% and droughts by about 7%. Now, the model didn't necessarily reproduce these impacts, and it needed some model improvement. That is, we improved the representation of crop varieties to better meet the locally observed growing season and the phenology of the crops in these particular regions, that allowed us to now reproduce the observed events and have these impacts in the model as well. So that's for the historical period, but the question is, how would these impacts change over time? There is one publication by Corey Lesk, who's a colleague at NASAGIS as well, one of his landmark publications back in 2016 that had a first glimpse of a time aspect looking at the global signal of cereal yields between the 1960, 1980, and then the 30 years after that, 20 years after that, and basically showed a doubling of impact. A PhD student of mine has done similar work for Europe and confirming that idea that the losses in cereals associated with these disaster years double or even tripled over the last 50 years. That paper is in revision currently and is hopefully going to be published soon. But the question is, how is this trajectory going to play out in the future? And to look at this question, we're using the AgMIP and GGCMI protocol for CMIP6, using PI control simulations and historical reference simulations to put future projections for RCP 2.6 and 8.5 into context, and especially the PI control run that is done for each climate model throughout the whole time period until 2100 really allows us then to speak to attribution questions. So the modeling results that I'm going to show after this slide are based on CMIP5 and LPJML only, because we just didn't have time to reprocess the new crop model simulations, and this will come in soon. So it's kind of a proof of concept, but I will guide you through that. So the first question is, we don't have the disaster database EMDAT that we used in the historical period to tell us what year was an extreme event or a disaster year. So we need to come up with a metric to identify those. And for drought occurrences, we use a 2 standard deviation threshold for historical anomalies, standardized anomalies to identify future droughts. So here for France, for instance, we see the 2 yellow shades for the 2 different RCPs show an increase in these transgressions of the 2 sigma metric. For China, same story for wheat. But in Russia, for instance, we see a trend. And that's not our own finding. This is in line with previous work. For instance, here, Newman et al. In 2018 showed a return period of droughts that significantly decreased in Southern Europe from a 1 in 100 year occurrence to less than 20 years under certain specific warming levels, SWL. But then for the Russian Federation, there's actually a decrease. So a decrease in return periods. So the drought metric indicates less events there. Now we can look at that as a frequency of droughts at country level globally compared to the historical period. Again, the yellow represents 2 future scenarios. We see a substantial increase in number, and the events per year across all countries, historically about 7.5. For reference, I included the EMDUD database. So we're, with our drought metric of the 2 standard deviations, somewhat in the same realm of the historically recorded disasters, is projected by the climate models to substantially go up in the future. This is done for maize, cropland, soil moisture in that top left corner, and for the other crops, it's a very similar pattern. Now, if we don't want to compare it to the historical period, but to the PI control run, so for the same time period done by the same model, we can actually speak to attribution, since the box plot illustration of the number of drought events for the 3 scenarios, PI control, RCP 2.6 and 8.5, don't overlap. In this case, it is very likely that they are statistically significant, which allows us to say that the increase in frequency is due to the climate change impact. Now, on the last few slides, I would like to actually look at yields, not just the events, but the impact on yields. And one of the working hypotheses here is that the yield variability, in this case, using the coefficient of variation, which is standard deviation divided by the mean, would increase in the future. And if we have increased variability, we're having more negative and more positive extreme years. And that's exactly what this plot shows. The yield change variability increases over time in the future. And that's also in line with previous work, showing the CV, the coefficient of variation globally, associating that with the idea of the higher risk of synchronized breadbasket failures. Now, I want to circle back on my last 2 slides on this composite figure idea, because that's where we want to get to. So now we need to talk about CO2 a little bit, because the crop models are hypersensitive to carbon dioxide increases. Not all of them in the same way, but this is where most of the model uncertainty comes from when we talk about impact or crop models. Now this figure shows a transient CO2 case and the bottom panel for constant CO2. Comparing these impacts for the worst 3 future drought years to the current reference period. Now this is not terribly informative, because it's it hides an underlying trend due to the CO2 imprint. So if we take out the CO2, the whole the time series shifts downwards. So this is the current reference impact. But what we are actually really interested in is the impact of the events compared to its neighboring years. So if the relative impact, the detrended impact, is getting worse over time. And indeed, if we look at these things in a detrended way, so we isolate the event signal based on the surrounding 6 neighboring years, it is indeed the fact that under higher warming scenarios, under RCP 2.6 and then further RCP 8.5, the impacts of droughts in this case lead to a higher yield impact globally across all major producing countries. For maize and soy as an example in this case, at the top panel shows the transient CO2 case and the bottom panel the constant CO2 case. So without CO2, the impacts become much stronger. But remember, this work is a proxy stand-in work done with LPJML. And on the first slide, we saw that LPJML is the most optimistic model in the whole ensemble of crop models. Plus it is CMIP5, which is more optimistic than CMIP6. So redoing all of this with the new crop model ensemble and CMIP6 data is likely to show a more adverse signal than we currently see here. Then real quick, I would close on this slide, putting it into the spatial context. The map on the left-hand side shows the historical impacts of droughts as simulated by the models at country level. And then for context, this is the relative change in drought impact at country level. And the interesting thing is that the spatial pattern of these impacts are not sensitive to the specific CO2 assumptions in our scenarios. The CO2 will affect the amplitude of things, but the geographic pattern, that is, droughts will become more severe in North America, much of Europe, Central Asia, South Asia, and China. That's a robust finding, and again, is likely to be more severe using the full ensemble. Just quick conclusions based on these preliminary results is that CMIP6 is revised downward, or the crop impacts are revised downward based on CMIP6. Climate change will significantly increase the frequency of droughts, and annual variability is going to increase with projected impacts on crops. And just as we've seen, the spatial pattern are robust irrespective of CO2 effects. Now, the next steps is to finalize the mean impact paper, the first 2 slides I showed for the IPCC AR6 deadline, and then we will come back to that extreme event evaluation without such a rush, and then run the full GGCMI crop model ensemble to redo all of these preliminary analyses. Then we will look at heat waves, which is hopefully and likely going to be a bit more straightforward than droughts. It is unclear in the literature if droughts really increase that much in frequency, but heat waves are much clearer. And then the final question we want to get at here is to look at if events, if we can identify events where droughts and heat waves occur at the same time in different breadbasket regions. So looking at that multiple breadbasket failure topic a little bit here too. With that, I would like to thank you for listening, and for the large team of people providing data and analyzing data, and really being the force behind this great community. And please let me know if you have questions.
Okay, thanks ever so much, Jonas. That was a really interesting talk. There's lots of information in that. We'll really need a couple of days to digest it all, I think. We now have 24 minutes before we have to go back into the main room and report back. And we've still got a 15-minute presentation to go. I know, Sergey, I can see you there. You saying, can you ask 2 very quick questions? I think if you can ask them in 30 seconds and if Jonas can reply in 30 seconds, then yes, you can.
Sorry, it took way too long. I didn't realize.
Thank you very much. I'm super quick. Actually, 3. One is, did you account for crop establishment dates? Is that something that is accounted for in the modeling work? Second question, what about the flowering stage? You mentioned phenology, but I believe that development stage when drought stress or heat stress occurs is critical. And then do you account for interaction between temperature and water stress? So these are very quick questions, but this is extremely important work, obviously. Thank you very much for the presentation.
Great. I think we can have another whole hour discussing these questions. So just real quick, and then we follow up sometime later. Planting dates, yes, are harmonized across all crop models, especially in this new simulation rounds. We have a new crop calendar product that is including new digitized observational data sets. So each crop model is asked to plant at a specific date in each grid cell. And then phenology is simulated by the model individually. We also prescribe the harvest date, but the model defines how to get there. So the model will tell you when anthesis is happening. We're not harmonizing anthesis dates. So each model has a different phenology approach. Regarding heat stress and drought stress, yes, all of the models differ between the 2, but models do much better when it comes to water stress than heat stress. So heat stress is actually a tricky aspect, and not many models, or not all models, have an elaborate heat stress mechanism. So yeah, this is kind of a can of worms where you really need to look at the details, and it's beyond the scope of this presentation to list all the different aspects of the models, but it is in field of active development to improve the physiological heat stress impacts. Not just the underlying water stress components, because that's pretty clear. But the heat stress is active development.
Okay, thanks ever so much, Jonas. I really want to give Bruce, who's waiting there very patiently, the chance to give his talk. Just before I hand it over to you, Bruce, yeah, great if you can start getting your slides already. I just want to point people to the very end of the Google presentation, where I've made a really bad attempt at starting to write some key conclusions so that we can be thinking about what we actually want to try to report back. So especially those people that have spoken, if you want to go into that section and try to edit those, that would be great. Okay, Bruce, over to you for some evapotranspiration.
Okay, thank you. Well, in the previous talk, where we heard a lot about what's going to happen with regard to drought and so forth, but what probably wasn't brought out is the uncertainty and how well the models that were being used to predict when drought was going to occur and how well they've been tested. And that's what this talk is going to be all about. Anyway, we got started about 4 years ago, and realizing that the crop growth models vary a lot in their predictions of the rate of evapotranspiration. Now, this talk is going to be about 42 different maize models and the ongoing intercomparison that we're doing. And yes, I've got 52 co-authors on this, and we're looking at 42 maize models. A couple of years ago, we got out a first paper on this where we did an intercomparison of 29 maize models, and you can look it up there in Ag and Forest Met. And I'm going to briefly go through the conclusions from that particular paper. First, we noticed that the ET values varied by about a factor of 4 in mid-season among the models when the first blind test. But then after we gave them all the data that they could, so the models were fully calibrated, they were still varying by about a factor of 2. There was little improvement in the early part of the season when it was mostly soil evaporation, but they did manage to improve their predictions when mid-season, when transpiration was dominant. I'm going to come back to this one, but anyway, much of that variability among the models appeared to be what they were using as their potential evapotranspiration. One thing that's been seen in a lot of these intercomparison studies is that the median of all the models is often a good predictor, but not all the time. When it comes to yield, several of the models were better than the median. And the best model for predicting yield was not even among the best 6 for predicting the seasonal evapotranspiration. One thing that came out was that there were several models where different flavors or different routines were used. And in this case, the DISET series maize model was run with first with an older Ritchie version for computing soil evaporation. However, we found in this particular test that the default that's in there now by Suleiman and Ritchie was actually poorer than the old one. So that was an interesting thing. And here's another case of that. The expert N model had 2 different flavors. There was a simple version that had a realistic root distribution, and then another detailed energy balance version that had the roots uniform with the total root sodium zone. Well, lo and behold, the simple version did better than the complicated energy balance version of predicting ET. There were many different approaches and complexity amongst the best models, and their accuracies were close to one another in many cases, even though they varied greatly in complexity. One thing we did notice that the models that are widely used were among the more successful, and that suggested that a larger number of users over very larger ranges of conditions and time had led to more testing and more improvements in models. Now, one thing that we discovered about 2/3 of the way through in that first intercomparison is that there must have been a water table present in that data set that was gathered at Ames, Iowa. And there were no deep soil moisture measurements to confirm or deny that. But looking at data from fields around, we concluded that, oh dear, that crop has to be getting more water than is coming from rainfall and storage at the beginning of the season. So that led us to move on to another intercomparison where we went out and found 2 data sets that we think are going to be much better, or they really are looking at much better. One's from Meade, Nebraska, where they're using eddy covariance. There were 12 crop years of data from there, 6 in a rain-fed field, and then another 6 in an irrigated field. And then in Bushland, Texas, they're using lysimeters, which is the gold standard for measuring evapotranspiration. And there are 8 crop years of data there. So we've got 20 different crop years of data to be comparison. Moving on here. The protocol for doing this intercomparison is that we started off with a blind phase. The modelers were furnished weather, management, soils, initial conditions, and phenology. And they got the results back by the end of February of this year. And then we went on to a second phase where I sent out to all the modelers the leaf area index data, biomass, and yields for only the fully irrigated treatments. And they got the results back by the end of September, just a couple of weeks ago. So what I'm going to be presenting is data that's come in mostly here from this phase 2. I've been analyzing data as fast as I could for the last couple of weeks. We have now started phase 3, and I sent out the evapotranspiration and soil moisture data for the fully irrigated treatments. Those are due back mid-December of this year. And then hopefully if that deadline is met, we'll move on to phase 4 and that'll get finished. And in that case, they'll get all of the data for all of the treatments, both well-watered and water stress treatments, rain-fed and 75%. So anyway, hopefully this will be done early next year. And I'll be analyzing data and we'll getting a paper out hopefully by this time of year from now. This is a list of models that are participating in round 2. And I think you'll see quite a few familiar names in there, as well as some I bet you've never heard of. I want to point out one thing here. Well, first of all, several of them have different flavors. For instance, this Sticks one has got a couple of different flavors. Maize Sim is a daily version and an hourly version. And then Distat Series Maize has got a whole bunch of flavors, and then the Distat XM. There are 2 maize models within the Distat umbrella. And the Distat group really got well organized, and there were 4 different methods. I told you there's a maize model and an XM model. Okay, within both of them, the potential evapotranspiration is being computed by 4 different methods. And then within that, there's the, as mentioned before, we're looking at this older Ritchie method for soil evaporation. And then the newer one, this is Ritchie et al. 2009, which is the Suleiman one. And then there's also this Hydrus one, and that uses the Richards equation to move water around in the soil based on water potential measurements. In all, there are 10 different flavors being used under series maize. So we can really, I think we'll be able to look at some of these mechanisms and subroutines within the model for that. Now, if we move on to the XM model, it's basically just the same as all of those, except we're not, I'm not doing it, except they're not doing the Hydrus version. So in all, there are 18 different flavors within the Distat family that are being intercompared. So I'm thinking that we'll be able to look at some of the details and what really works there. Okay, moving on. This is the field NE2 that's in Meade, Nebraska. It's got a center pivot irrigation system, and that's where the data are coming from Nebraska for the irrigated field. The rain-fed field doesn't have the irrigation system, and it's nearby, and I don't have a picture of that. Here's a picture of the one field there in Meade, Nebraska, where I visited back last year. And this is the flux tower where they're measuring the evapotranspiration. And then back here is what they call the radiation tower with all the radiation sensors on it. It's painted black to reduce reflections. The other field is in Bushland, Texas, and they've got a weighing lysimeter there. It's a Texas-sized lysimeter. It's 4 meters on the side. So in this particular, on the side, and then in this slide, you can see it holds 4 rows of cotton. And that would be the same if it were maize there. But, so you can get some idea. And back there in the back is one of their irrigation systems. They call it MESA, which stands for Mid-Elevation System Application, I think it is. Okay, here's some actual data. This is from the Bushland site in 2013, where they were providing 100% of their calculated evapotranspiration. So this is basically getting well watered. And I think most of you are familiar with box plots, but basically these green boxes here that are there for every day during the growing season, that's where 25% to 75% of the model's results came in. The whiskers go from 5% to 95%, and then these dots are the outliers. And you can see the outliers go from being really low to really high. So this is kind of the same picture that we got in terms of the total variability that we observed in round one of this with the Ames data. However, if you start looking more closely, and well, you can't see it, but I could see it looking back from before. The green bars here, the boxes, are actually smaller than they were in the previous data. And if you look here, the blue curve is what the median is, and then the orange or the reddish one is where is the observations. And as we go along here, beginning of the season, they're really not doing too badly until we get some really high ET days. And then the models generally are just not picking up that high, those high ET rates. And that's back here when there was mostly soil evaporation, and then we're getting to mid-season, the same thing was going on. A lot of the times it wasn't doing too badly. We got towards the end of the season and the real plants were shutting down, but the simulated ones were still cruising along here and transpiring. Now, if we go to another field there in Bushland, that was receiving 75% of what their calculated ET was being. So this one was getting some water stress. And if you go on here, the same story early on, and then we get into mid-season, and lo and behold, the simulated ones are actually doing a better job of getting close to what the median's closer to what the observations are here. And then at the end of the season, the same story again. Observed plants are shutting down, but the simulated ones are still transpiring. I just grabbed 6, or actually 5 plots kind of to give you a range. They're not random, just to give you an idea of what kind of variability I was seeing out there in the reported data. This is the daily ET through the season. This is from Mead. And here's the median, fairly respected. Here's one that looks like it's doing better than the median. And then we got one over here that really did quite poorly. Okay. This is the normalized root mean square error of daily ET rates. And the top is the 100% irrigation, and the bottom is the 75% irrigation treatment. And this is for all the different models. I assigned model numbers going from 41 through 82 in order to make sure that they were all unique and weren't overlapping the round one work. The median is off here on the right. And so you can see that the median and some of the better ones here on the 75% one were, and also up here, if you look up, they're about, you look at this, that's about 3/10 or 30% of variability then compared to what the observed mean for all of this was that was used for the normal visitization. But there are a lot of some wilder stuff going on here with the 75% one. Any other points I should make here? I guess that'll do it since we're running short. Okay. Then this is observed versus simulated cumulative ET from -10 to +20 days after planting. So this is pretty much bare soil and soil E dominant for this period. You can see the median was just really pretty well nailing it. You look over here and some of them are pretty far off. This one was really underestimating what was going on. This one was simulated, was overestimating, and you can get some idea of the range of what the models were producing for this evaporations part of it. Now, if we move on to +41 to 100 days after planting, this is be dominated by transpiration. And this is again for all the 20 treatments. The median isn't doing too badly. Here's one that looks like it's doing pretty good most of the time, but it really missed it for two of the things. Actually, two dots there close together. Really missed it for two of the cases. Here's one's doing pretty well. Here's one that's way off on the slope. So that gives you some idea of how well they're doing as in the data I'm seeing. Now here's a plot.
Bruce, can I just, sorry, I'm just aware we've got to go back in 3 minutes time. So if there's any way you can maybe back up in a minute, so we've got just a couple of seconds to think about our key conclusions, that would be great. Thank you, sorry.
Okay, I'm going to jump to this one. This one shows the box plots then for all the 20 different treatments. If you look at longer lengths of periods here, the models are not doing too badly compared to the daily stuff that was getting on. This is the soil down here and the transpiration part of it up there. Basically, conclusions that we've gotten so far is that several models are doing a respectable job. The others are doing rather poorly, especially on the daily stuff. The median generally does, but not always, is a good predictor. They're certainly doing a lot better with cumulative ET than they are with the daily. And there's a lot more analyses and 2 more phases remain to be done. And so I guess I better stop right there. Thanks for your attention.
Okay, thank you ever so much, Bruce. I'm really sorry to have rushed you a little bit there at the end. I mean, just a comment from me, it's interesting because there's an awful lot of stuff that we could be doing in terms of ozone flux when you have that evapotranspiration data. I'm just looking at some of your sites and thinking if you could stick some ozone monitors out there as well, that would be absolutely amazing. But that's my own personal opinion. So I'm not sure we've got, if there's any burning questions, then maybe we can put them in, people could put them in the chat. What I'd really like to do, we've got 1 minute left, is just if everybody could have a look at these key conclusions, maybe if I can just share my screen again really quickly and just see if people are sort of relatively happy with these. Don't look at what you're seeing at the moment. Let's bring this up here. So everybody should be able to get access into these last 2 slides of this PowerPoint presentation. And I've just had a go at trying to pull together some thoughts. So for example, Jonas, there's one here where you were talking and I was trying to write down some key aspects of what you might have been talking about. If you could sort of go in there and have a think and see if there are things that you might want to add, that would be fantastic. Just, I'll run through these. So this first one is really just thinking about this yield gap and sort of thinking about the yield constraints that I mentioned right at the top. So are there ways in which we can sort of get a better understanding of which of the stresses which are most contributing to the yield gap? Is that a useful thing to do? Looks like somebody's added another one in here. Many stresses on plants. We're only currently exploring a subset. great need to add stress response from climatic and biotic factors with particular factors, very important in some regions and farming systems. So that could be a research agenda, allowing us to increasingly look at connected extremes and interactions between long-term pressures and short-term shocks. Brilliant. I've just added in here my one about the COVID-19 from a pollution point of view, and that might provide opportunities to test some of our models. Again, another one about stress interactions. Can we use all the knowledge? I mean, you know, just the talk that Bruce just gave then about the in-depth knowledge about evapotranspiration that exists there, that could be useful in terms of us getting a better understanding of ozone effects and mechanisms. We've got a thing here about pests and diseases, so collectively causing yield reductions in the range of 5 to 40%. So we need to bring that knowledge into crop models, otherwise they're going to be overestimating yields. And then this one that was, I tried to bring together some of the information that Jonas provided, sort of thinking about these effects of the extreme heat events. What's going to happen in the future? What sort of metrics should we be using? And what are the implications in terms of changes in yield variability? Any?
Looks good.
Good. So I think we probably have to shoot back in unless there's any burning comments in response to this. If not, then I'll just quickly say thank you ever so much to the speakers who all gave brilliant talks. Thanks to all the really good questions that we had. And thanks also to people that have started to fill out post-it notes here. So you've all got access to this, so you can sort of get a bit more information. Okay, I think we should go back into the main room. So I will see you there. I hope you all have, shall I just quickly put the link in the chat? That might be a useful thing to do. I'm hoping it's going to be the same link. And then you should be able to, let me do that to everybody. Everyone, use that to get back into the main meeting, hopefully. Okay, see you soon.